So we are talking about the scrotum, and we're talking about how the scrotum can regulate temperature to maintain an optimal temperature of about 35 degrees. Okay, so we need to regulate temperature, and we know that body temperature is 37 degrees centigrade, but optimal sperm cell temperature is closer to 35 degrees centigrade. That 2-degree difference is a big deal. So we have to be able to maintain that, and we do this through 3 different mechanisms. So 3 mechanisms to adjust the temperature. And 2 of these mechanisms are related to muscle function, and one of them is related to vascular function. So one muscle that is present in the scrotal tissue is a muscle called the Creemaster muscle. Now, the Creemaster muscle, we're ahead of ourselves, I think. Yeah, there we go. Creemaster muscle is this muscle that descends along the spermatic cord. And so when this muscle contracts, it's actually going to lift or elevate the testicular tissue closer to the body. And when it relaxes, it will allow it to descend lower. So that Creemaster muscle, when we have a drop in temperature, because if we go below 35 degrees, that's not optimal either. We want to maintain 35 degrees. That's our homeostatic set point. So if there's a decrease in testicular temperature, the Creemaster muscle is signaled to contract, and this allows the testes to move closer to the core of the body. We're going to call that the oven. So we put the buns in the oven. When temperature increases, those Creemaster muscles will relax. So they're triggered to no longer be in a contracted state, and this allows the testes to descend further from the core body temperature.
Is that particular area of the feeling much warmer than any other part? Like that area in there? I mean, like, is it warmer than like the rest of the body?
Here, like where we're adjusting the
just all of like that whole area. Is the whole area?
No, this is colder because it's maintained at 35 degrees centigrade. The core body temperature up here in the core is 37 degrees. But if we move the testicles closer to where it's warmer, they're going to gain some of that heat. And if we move them further away, they're going to lose or not gain that heat. Yes, JC.
How much of an influence does like your abdominal muscles have on the Creemaster muscle?
Very little influence. The Creemaster muscle is not really attached. So I mean, you're asking if you flex your stomach, can you cause your testicles to jump, I guess? The answer is not really. This is a pretty independent muscle. And it's important that it's independent because we want to maintain that 35 degrees. And we want the mechanism to do it when it's supposed to do it and not influenced by any other muscles. Great question. Okay, so that's just one of the muscles. And again, these are the fibers of that muscle descending down along the spermatic cord. So this is our spermatic cord on this side. We have the muscles that have been removed. They would lay on top of and around the spermatic cord, and they contract, hold it up. Then we also have a muscle down here in the actual sac itself, and this is called the Duct muscle. In the Duct muscle, this is going to be smooth muscle. So this is not attached to bones or other organs. It's just smooth muscle, and it's near the surface of the skin. And what the dartos muscle does is it allows the scrotal sac to actually change its tension. So this is going to cradle both of the testicles. And so when it's cold, the dartos muscle will contract. And so by cradling the testes in the scrotal sac, when it contracts, it pulls up on that tissue, tightens it up, which closes the gap to the body. So it brings it close to the body. Same kind of concept here. We bring it closer to our core heat production, and it's going to gain heat from being closer to the core body temperature. But there's a second thing that happens as well. By moving the scrotal sac closer and tightening it up, we reduce surface area. So scrotal surface area is reduced. And by reducing the surface area, we can actually decrease the potential for heat loss. So the heat loss is reduced because there's not as much surface area for heat to be removed. So that's what it is going to look like in the cold state. All of this undoes in a warm state. So if we're starting to increase above 35 degrees, dartos muscle along with the Creemaster muscle will both relax. All right, now the last, you can see there's a lot of vasculature. This tissue here, it's a cord. It's wrapped connective tissue. There's a layer of fat in there, and then loaded in there is a capillary network. This capillary network is called the pampiform plexus. So it's the pampiform plexus, and that blood supply is going to travel down the spermatic cord and interact with the testicular tissue, and then we're going to have an artery that enters and a vein that leaves. The vein that is leaving the plexus So the vein leaving the plexus runs alongside of the artery that's coming in. All right, so I'm going to try to draw this out in a little bit different detail. So we'll go red here. Hopefully you can sort of see that. That's artery coming in. I don't know if you'll be able to see blue or not. Yeah, it looks okay, right? You can see that. The artery and the vein entering in and leaving. So this is where testicular tissue is going to be. We have blood supply coming in and then blood supply leaving. The blood supply coming in collects the heat. It's going to pull heat off of the bloodstream. And so the venous blood that's leaving is going to be quite a bit warmer. The arterial blood coming in is quite a bit cooler. And so we're actually going to have a countercurrent exchange system where heat is exchanged. Other direction. Let's try that again. So the blood comes in. This is 37 degrees, right? So it's coming in warm, and as it enters into the scrotal sac, we have heat that permeates away from the testicular tissue, dartos muscle, and all that. So that 37-degree temperature is coming in. The blood leaving is cooler, 35 degrees or below. That's leaving. So as the blood's coming in, heat's being pulled away, reducing blood temperature here, countercurrent exchange in the other direction, where that heat coming in gets picked up by the 35-degree temperature. So the blood that's coming into the testicular tissue is going to be reduced because of that countercurrent mechanism. So the vein plexus is in the cord, and it runs alongside of the artery, so it contacts artery, and then through that countercurrent exchange mechanism, we can reduce the heat of the blood coming in from the body. So the 37-degree body temperature blood gets reduced because of the countercurrent exchange, because we've already reduced the venous blood coming back. So combine these three mechanisms come together, and they help to maintain a very consistent 35-degree temperature. I see a lot of confused faces. What word are we?
Is there, like, heat like coming down and then the air is coming down?
Heat's just radiating off because temperature out here is cooler. Body temperature is 37 degrees, room temperature is 25 degrees.
So when it goes out, it's going to be hot, that's cooler because the hotter air comes in.
Okay, so 37 degrees here, we go through countercurrent exchange. This is going to be closer to 35 degrees as it enters into the testicular tissue. Heat is dissipated, so the blood that comes back out is actually closer to 35 degrees here. And if I were to change the picture, we'd probably be closer to 37 degrees here. So blood comes in at 37 degrees and exchanges with the cooler blood here, that's 35 degrees, moving in this direction, 37 to 35. Does that make sense? We're good here. All right, so moving away from the testicular tissue, we have the ductwork. It begins with the epididymis that leads into the seminiferous tubule. And really, for all practical purposes, you can see that the epididymis is really coiled up, additional coiled up tubing that we would also find inside of the testicular tissue. So we're going to begin here with the seminiferous tubule that makes up the testicular tissue and the epididymis, and then we'll lead into the vas deferens. Actually, let's stay on that last slide so we can reference that picture. Okay, so within the testicle, within the epididymis, we have coils called seminiferous tubules. This is the actual location of spermatogenesis. So these are the tubes where sperm is made. We'll find them contained in the testes, and then they join to form the epididymis. Now, mostly spermatogenesis is occurring within the testicular tissue, and the epididymis is just basically a site of additional storage and some sperm maturation. So within the testes, the seminiferous tubule, you'll have two tubules wrapped all up inside of the seminiferous tubule, wrapped around each other. And then as we enter into the epididymis, the epididymis is going to be the tube that's outside the testes. It is a single coiled duct. And it is the tube, you can see it's a single coiled duct here, and it leads into the vas deferens, or the ductus deferens. So it leads to the vas deferens, or also called the ductus deferens. Spermatogenesis is primarily occurring within the seminiferous tubules that we find bound in the testes. The epididymis is going to be a storage site for sperm, the reproductive, male reproductive gamete, and it's going to be stored there until it's used, which is ejaculation. So leading away from the epididymis is the vas deferens, or the ductus deferens. So here's testy epididymis, and then this is our vas deferens, also known as ductus deferens. This is basically just a tube that's going to transport sperm from the epididymis, and it's going to transport the sperm into this region of the male reproductive anatomy. There are several glands that are associated with, or that are found rather, in this region of the male reproductive anatomy, just set below the bladder. So the vas deferens, or ductus deferens, leads to the seminal vesicle, and we'll talk a little bit about that in just a second, and then a duct called the ejaculatory duct. Now, there is going to be, in addition to the transportation function of the vas deferens, some additional storage of sperm. Not near as much as we would find in the epididymis and in the testicle itself. Now you do have, or males have, two vas deferens that converge. They bifurcate right here inside, which is analogous to this structure here, inside of, with the seminal vesicle and the prostate gland, and then there's another gland there called the bulbourethral gland and the ejaculatory duct. Okay, so the seminal vesicles, which is going to be the end of the ductus deferens, are going to pass the sperm. These are basically the points of bifurcation, or convergence, I should say, of both the vas deferens through into a common duct, which is going to be the urethra. Now we're going to talk just a little bit about sperm survival. I started this whole conversation saying that successful reproduction, fertilization of the ovum, is basically an impossible feat. And so we have to do a large number of things to make it more probable, like maintaining an optimal temperature so that we can get a maximal production of sperm. We're going to come back and we're going to talk about sperm survival during this process of ejaculation in just a few minutes. But kind of looking ahead, sperm is the male sexual gamete, and it is going to be deposited into a fluid called seminal fluid, or indocin. And it's going to be these glands here that generate and produce that fluid. So we'll come back and we'll take a look at those. So not only does the seminal vesicle pass the sperm to the urethra, it's also going to induce smooth muscle contraction. Okay, that'll facilitate movement of the sperm. Provides a rhythmic motion, or a rhythmic contraction. It's going to add in ingredients to make semen and pass all of that into the urethra, which is going to be located in the external genitalia. Which includes both the scrotum and the testes, and then also the penis. Now, the penis is going to be very important to maintain an open urethra. For sperm delivery. If there's any sort of resistance in the urethra, that resistance pushes back on that sperm delivery, further reducing the probability that fertilization can actually occur. So erectile tissue in the penis is going to primarily be there to help maintain that open urethra so that flow of the sperm cells and semen during copulation is going to be possible, or increasingly possible. Okay, so that's going to be the erectile tissue. The erectile tissue, we have the two structures on either side of the penis, which are the corpus cavernosum, and then so that's on either side. And then along the top, there's another erectile tissue called the corpus sponginosum. So corpus cavernosum, corpus sponginosum, to maintain the open urethra. Okay, so let's move forward to sperm production. So this sperm production, also referred to as spermatogenesis, we're going to get there in just a second. Anatomically, this is going to occur in the seminiferous tubule. And what we're going to find out, so this is a cross-section, an electromicrograph, scanning electromicrograph cross-section of seminiferous tubule. And what you can see is you have all of these cells that are, it's a false color image. This isn't what it really is. It's not purple. It's just a highlight. Make sure that that's more visible. So each of these, you can see these are individual cells. Some of the cells are cells that eventually will become sperm cells. Other cells are going to be support cells. So there's going to be three types of cells that we're going to find in here. And then down here in yellow are the mature sperm. So this is a tiny little cross section here. And already, I mean, you can try to go through and count, and there are already hundreds of thousands of sperm cells there. So take that over the length of the seminiferous tubule. Normal fertility is about 100 million sperm available at any given time. And those are replenished on a frequent basis. This is so this was the scanning electron microscope. This is just a light microscope with staining to highlight. You can see that we have different types of cells. There's connective tissue. This is the lumen. You can see some of the flagella of some of those cells. The epithelial covering, and then the three types of cells that are going to be present. You're going to have the cells that give rise to sperm, which really there's going to be a variety of those because we go through differentiation, and we start out with a primordial sperm cell, and eventually we differentiate to a mature sperm cell. But then there's going to be two additional cells, Sertoli cells and interstitial cells. The interstitial cells are also known as lighting cells. So three types of cells are going to be present. the Sertoli cells, and these are going to be the cells that support sperm development. So you should be thinking in terms of nutrient provision and waste removal. We're going to have the actual sperm cells themselves. And again, this is going to be a variety of different cells, from the primordial spermatogonia through the mature sperm, and I guess about 7 or 8 intervene cells. And then we have the interstitial or the Leydig cells.
What's that?
Development.
No.
You got that one, but there's another word that you couldn't get?
The right before cells. Like, what is it? Like Sertoli.
Sertoli.
Okay, that's not written down. That's not much.
Creepy. Leydig. Leydig cells, also known as interstitial cells. And these are actually going to be the cells that generate everybody's favorite hormone, testosterone. And testosterone is really going to be involved in regulating the rate by which spermatogenesis occurs. Okay, sperm production properly is going to be referred to as not even close. Let's try that again. Okay, so spermatogenesis. All right, hopefully you're beginning to recognize a lot here, and you should be able to identify vas deferens, epididymis, the testicle with its seminiferous tubule. This is a cross-section of the seminiferous tubule. Here's our connective tissue layer, and then throughout here we have those 3 types of cells. We have all the different types of sperm cells, spermatogonium, spermatocytes, mature sperms, all are going to be present in there. And then we're also going to have the supporting cells, the Sertoli cells, and then the Leydig or interstitial cells. So this process of spermatogenesis, as you can see in this figure here, it's basically going to be well-regulated sequences of mitosis and meiosis. So this involves a series of cellular divisions. And in our reality, the series is going to start out with mitosis. So what is mitosis going to do in terms of chromosome number? Is it going to reduce it, increase it, or help, or keep it remaining the same? It remains the same. So 46 chromosomes enter with one cell, 46 chromosomes. At the end of mitosis, we doubled the cells and then split them, so now we have 2 cells, each with 46 chromosomes. That's going to be followed up with meiosis 1 and meiosis 2. At the end of meiosis 1, we'll have 4 cells that contain 46 chromosomes. At the end of meiosis 2, we'll have 8 cells that contain 23 chromosomes. So each step along the way of mitosis, meiosis 1, meiosis 2, each step, we are going to have differentiation of the cells. The cells are going to become differentiated. They're going to become a new type of cell. Now, this process of going from mitosis to meiosis 1 to meiosis 2, it can be slow or it can be relatively fast. And that rate The rate of the control for rate is going to be testosterone. So prior to puberty, there's no perceptible levels of testosterone or low perceptible levels of testosterone at puberty. The Leydig cells, interstitial cells begin to produce testosterone in response to the pituitary signal, which is going to be luteinizing hormone, the follicle stimulating hormone. Testosterone levels beginning to go through a pulse. Ladies, you go through a cycle every 28 days. Guys go through a cycle every 24 hours. Maybe it explains everything, right? In that pulsatile 24-hour sequence is pulsing testosterone, so that we have this increase in production of sperm cells to maintain 100 million available when they are needed. So, in all reality, I've given you a really brief annotated view here. I am going to make you responsible to take a look at this and to really figure out cell names, to identify what's happening at each step along the way. Mitosis, meiosis 1, meiosis 2. Mitosis, we have one cell with 46 chromosomes, so we'll go cell here, we'll go chromosome here. At mitosis, we go from 1 with 46 to 2 with 46. The cell type here is going to be a spermatogonium. Let's even go ahead and maybe increase the picture size just a little bit.
You said the one cell 46 chromosome was mitosis.
So you start out with this thing called a primordial germ cell that's going to differentiate into this cell that through mitosis becomes a spermatogonium. Okay, so we start with this is going to be our germ cell, our primordial germ cell. It undergoes mitosis. Mitosis happens right here to give us rise to 2 46 chromosome containing spermatogonium. Those 2 cells, which is not really shown here, notice that we're basically just tracking one cell at a time. We actually should have this figure duplicated right here as well, because those spermatogonium, they are going to differentiate into a primary spermatocyte. That primary spermatocyte, there's going to be 2 of them with 46 chromosomes. They'll undergo meiosis 1. Meiosis 1 is going to give rise to our secondary spermatocytes. There's going to be 4 of those, each with 46 chromosomes. Okay, so these are my primary to secondary spermatocyte. Then we're going to have our second mitosis, mitosis 2, and our 4 46 containing chromosomes are going to be each of them split, no intervening DNA synthesis. So 8 cells becomes, the 4 46 containing cells become 8 23 containing cells. This is going to be what's known as a spermatid. The spermatid will go through a process known as sperm maturation. So these eventually will become the mature sperm that are going to carry the 23 chromosomes and have all the other required ingredients for successful fertilization. Okay, so we get our mature sperm, and those mature sperm have to survive. They have to survive initially in the seminiferous tubule, but they really need to survive when they are utilized. Does everybody have all of this? Is everybody golden? Now, sperm survival is going to be very important. And the reason that it's going to be very important here is because this is going to be one of the points that really significantly reduces the probability of fertilization occurring. If you really step back and think about it, these sperm, they have to go through a relatively long journey. They're going to be interacting with a hostile environment because that environment is maintained to be slightly acidic, to be protected from microorganisms. They also have to find the one ovum that's been released and it's got a 50-50 chance it can either be released from the left ovary or from the right ovary. And we're going to find out that usually what happens is it alternates. That ovulation occurs on one side 1 month and then occurs on the other side on the other month. So it's a moving target. But there's a lot of cool things that we do that help to facilitate this process and make the impossible highly probable, or at least probable. Okay, so sperm, when it is going to be used, it has to be deposited into a fluid. So it's going to be contained in a fluid that is called semen. And actually the semen, as we move along the reproductive ductwork, it's going to change in composition as it goes through certain glands. So the composition is going to change. Semen is initially produced in the seminal vesicle. So the seminal vesicles are going to be that initial point of semen production, and it is initially produced as seminal fluid. So the seminal vesicles secrete this solution called seminal fluid. And that seminal fluid is going to be comprised of a bunch of different ingredients, 2 of them that end up being really important. One of those ingredients is fructose. So this is a sugar. Once the sperm are released from storage, they no longer have access to a nutrient source. So the seminal fluid is actually going to give them their bagged lunch that's going to be available as they're traveling along. So the fructose acts as an energy source for those sperm cells as they travel through the male ductwork into the female ductwork. A second chemical that's going to be present in the seminal fluid is a chemical called prostaglandins. Prostaglandins. Now, if you know anything about prostaglandins, you'll know that prostaglandin can actually be used during the birthing process to cause uterine contraction. Prostaglandins are put into the semen from the seminal fluid. And what the prostaglandins do, this is so crazy, it's all about making the probability more likely. The prostaglandins are actually going to act on the female. And they stimulate female contraction. And what happens is female contraction is stimulated. This allows the semen containing the sperm to be moved up from the vaginal canal through the cervix into the uterus. Now here's where it gets really crazy. I just told you that the uterus and the fallopian tubes with their associated ovaries it's a uterus. Okay, there's the vaginal canal. So semen come up, and fertilization is going to occur up here in the oviduct. Now, let's say 100 million sperm have just been sent up, and they are traveling along. And it would be really great if most of the sperm cells ended up where the ovum was just ovulated from. So we want to go towards the ovary that has just produced, or it's just ovulated the ovum, right? That would be great, right? So prostaglandins, in the presence of prostaglandins, and how it works, we really still don't entirely understand, but it seems like there must be some receptors that get expressed at the point of ovulation only on the side of the uterus that's released the ovum. So here is month 1, it's released on this side. Month 2, it's released on this side. Probability, we just say that 50% would go this way, 50% would go this way. But what we actually now understand is that if this is where we've just released the ovum, 95% or 90% or more of the sperm end up traveling up that uterine tube, that oviduct, making the chances of fertilization just that much more significant. So provides efficient movement, and really it's not only just efficient movement, it's also guided movement. So efficient guided movement. So this is just sperm getting dissolved into seminal fluid. The seminal fluid, let's see if I can bring up that's an ovary, not what I want. Let's go back here. So sperm cells come along right here in the seminal vesicle. They're now mixed with that fructose prostaglandin containing solution called seminal fluid. The next part of the reproductive tract here, you can see urinary bladder. This is going to be the start of the urethra. Here's our bifurcation. Right before that bifurcation, this is the prostate gland. Okay, so the prostate gland will be our next gland that the seminal fluid is going to interact with. Now, I've already told you that you have the acid mantle that covers your skin and goes up into all of your openings, making everything slightly acidic. Being slightly acidic is protective against microbes, but it's not very conducive for survival of cells that are not your own cells. So the prostate gland is going to mix into that seminal fluid an alkaline fluid. And that alkaline fluid, this is a base, opposite of acid. It's going to neutralize the acidic, protective vaginal environment. So that alkaline fluid gets added in. It's responsible to neutralize the acidic, protective vaginal environment. So in terms of pH, the pH is normally around 4 in the vaginal canal. And the pH is going to rise from that 4, which is protective, to around 6, which we're going to find is actually optimal. Let's try that over. It's going to be optimal for sperm and for sperm survival. Now it's fleeting. That pH is only going to increase to 6 for a short amount of time, and then we'll drift back down to 4, again, be protective. After the prostate gland, we have this last little gland. It's a pretty weird named gland. It's called the bulbourethral gland. And we actually don't mix anything new into the seminal fluid. What's actually going to happen is the bulbourethral gland is going to send out its secretion right before the semen crosses through that portion of the tube. Okay, so what is it actually going to secrete? It secretes mucus, which coats the urethra. And it's going to provide two things. Secreting the mucus provides two things. One is it washes away any acid residue left over from urine, since it is a shared tube between the urinary system and the male reproductive system. So we're going to wash away that acid. That mucus is also going to provide lubrication. Which will reduce the resistance to flow. and will optimize delivery of most of the sperm that were initially ejaculated. So all of these things, changes in temperature, regulating the temperature very, very rigidly. The amount of production, just 100 million sperm that are being produced and maintained in a normal, vigorous male. And then all of the changes to seminal fluid and the seminal fluid increase the improbable to probable.
So all of this happens.
All of it. Constantly.
So it happens like, you said constantly, so it happens like every single day.
Not all of it, because really the prostate gland and the seminal vesicle are they're only producing semen when it's occurring, when sex happens. When you make it have sex. But the sperm cells, all cells have a lifespan, right? We know things like red blood cells, 120 days. Sperm cells, about 9 weeks. But it's not like every 9 weeks it's like, okay, new 100 million sperm. It's constantly happening. Each individual sperm is someplace within the process of maturing and going through spermatogenesis. So it's constantly recharging that 100 million supply. Now, about 100 million of them are going to be delivered during sex. And so about 3 days later, you're fully recharged. Okay, so you have 100 million sperm. Yep, it takes about 3 days. So to optimize male fertility, this is really crazy too, and I'm not sure why this happens, but married couples, how frequently does the average married couple have sex? Every 3 days. And so that means that most couples, even above and beyond 100,000 sperm and all of this other stuff that's going on, most couples are having intercourse when the male is most fertile.
So if we get between them.
Low sperm count. Low sperm, and it reduces the likelihood. Pretty crazy, isn't it? I guess we'll stop there. We'll pick up a female reproductive system on Friday.